Hello everyone. So my name is Jason Nomberg. I'm a first year PhD student in the virology program at Harvard. So my talk today is about viruses as medicine. Now, oftentimes you think viruses are associated with disease. Viruses make you sick. And for most of human history, that has been how humans have been involved with viruses. Now, viruses also are great tools to use that we can use for medicine. So that is the basis of my talk. So let's begin. So viruses can be used as tools to solve urgent problems. Now, to start my talk, I will introduce you to viruses, evolution, and the evolutionary arms race. These are very important concepts. And next, I will explain how can viruses be leveraged to address two critical biological problems. Now, there's a plethora of problems that we can address using viruses, but I've chosen for this talk just to speak on antibiotic resistance as well as cancer. So we'll begin with my introduction here. So what are viruses? Now, viruses are extremely tiny. They exist at the boundary of what is considered alive. To explain, viruses are made up of proteins and genetic material, and they can also have an outer membrane. Now, this is an artist's rendition of influenza virus. Everyone's familiar with the flu. You can see on the inside you have, let's say a genetic material. On the outside you have protein parts, so virus parts. Now, viruses depend on a host to copy themselves. So they depend on a cell to copy themselves. Now, normally, as I mentioned, they're considered pathogens, but what if we could use them as tools? So, just to begin, how do viruses work? Now, if you have a virus here and you have a cell, so this could be a human cell, this could be really any virus susceptible cell. The virus must get into the cell. Now, when the virus is into the cell, its genetic material is somehow released. Many viruses, so this whole life cycle that I'm going to show to you today, many viruses have different twists and turns on this life cycle. Now, the virus genetic material is released, and this genetic material can be used to create virus parts. Now, these virus parts can go back and use the virus genetic material to make even more virus genome, which can make more virus parts. And soon you have just, a cell becomes just a factory for replicating virus. And at a certain point, some viruses function by killing the cell and just releasing all of this virus. Now note, there is one virus in, there's many virus out. These viruses can go on to infect other cells. So this is how a virus propagates itself. Now, so I kind of have to comment on this for my talk, but how does life evolve? So there's a thing in molecular biology called the central dogma. You start with DNA, you get RNA, and RNA codes for protein. DNA is the genetic material I'm sure you all are familiar with at some point. Now, on occasion, there can be mutations, or there can be changes in the DNA. This can be caused by various things. Changes in DNA reflect changes in protein. Changes in DNA reflect changes in RNA, which code for changes in a protein. Now, if you have a protein here, this blue square, and you have a change, and suddenly it's a new, it has a modification. Now it has a red outline. Let's see how this reflects on evolution. Let's say you have a pool of cells. These are all individual cells. This one cell had a change, and now its blue square has a red background. Let's imagine that there is a virus introduced to these cells. And let's say the virus can only kill cells that don't have a red background. These cells will die, and this cell with the change will go on to live. Over time, all of the cells will become like this. Now, so I brought that up because it's important to realize what causes evolutionary change. So there is an evolutionary arms race between humans and human pathogens. Let's say there's a cell here and there's a virus trying to get into the cell. The cell does whatever it can. It will evolve such that it's trying to restrict the virus from being able to get into the cell. But over time, the virus develops a new way of getting into the cell. This is just evolution. The viruses that can't get into the cells will die off. The viruses that can get into the cells will make more copies of themselves. But over time, again, the cells that are more successful in resisting the virus will go on to make more progeny. So it's a back and forth. It's an evolutionary arms race. Now, let me talk about bacteria briefly. So bacteria are much larger than viruses. Bacteria can replicate themselves on their own. Now, bacteria are also in an evolutionary arms race with humans as well as with antibiotics. Now, let's imagine we have bacteria and we can treat them with some antibiotic that will kill them. Over time, bacteria that can resist the antibiotics in some way will be the ones that will copy themselves. And soon, bacteria are resistant to these antibiotics. So this is a concept called antibiotic resistance. And that's the first problem that I will speak of that we can address with viruses. So antibiotics are used to combat bacterial infections. They kill bacteria by interfering with bacterial-specific processes. So things that bacteria do that humans don't do, or maybe differences. Over time, bacteria that are better at surviving antibiotics will go on to make more copies of themselves than the bacteria that die off from the antibiotics. So let me just kind of show you over time how fast antibiotic resistance emerges. So in 1950, tetracycline, which is an antibiotic, was introduced to the clinic. Just 9 years later, we isolated tetracycline-resistant Shigella. In 1960, methicillin was introduced. In 1962, we identified methicillin-resistant Staphylococcus. In 1996, levofloxacin was introduced. In that same year, we isolated resistant bacteria. 2010, again. 2011, resistance. Now, I'm sure you've all heard of penicillin. This was introduced to the clinic in 1943. But even before we used it in the clinic, we were aware of penicillin resistance in Staphylococcus. So antibiotic resistance is rapid. Now, one potential tool we can use against bacteria are phage. Phage are viruses that affect bacteria. They make up the majority of all organisms on Earth. So just to put it in context, there's 8 billion people, and there's about 10 to the 30th phage. That's just an estimate. There's so many phage. And phage have evolved and adapted to infect and kill bacteria. So remember, just like a bacterial cell is very similar to, this infection is very similar to a eukaryotic virus. So the phage goes into the bacteria, makes a bunch of copies of itself, kills the bacteria, and the phage go on. So phage therapy has a variety of benefits over antibiotics. So I'm going to kind of mention one. Now, normally with an antibiotic, you can do combo, you can give people multiple antibiotics, but generally there will be resistance. So let's just kind of discuss phage. So when you treat an infection with diverse batches of phage, you can help prevent resistance. So I'm just going to annotate this. Imagine you have a phage that has some, it has some requirements for entry. And this is a bacteria here, and you have some protein on the bacterial surface. This protein has 3 different parts. It has a yellow part, a green part, and a blue part. The bacteria needs this protein, so it can't get rid of the protein altogether. And the phage needs this yellow part of the protein so that it can get in and kill the bacteria. The thing is, over time, bacteria that spontaneously make a change in the orange part of this protein will prevent this phage from getting in. So over time, the bacteria will become resistant to this virus. One of the interesting things we can do with phage, though, is we can treat an infection with multiple different phage against multiple parts of a protein. So instead of just treating a bacteria with phage against the orange part, we can treat them at the same time with phage against different parts of this protein. Now, it is very difficult for this bacteria to make so many changes that can prevent all of the phage from killing them. In addition, unlike antibiotics, phage treatment can target disease-causing bacteria while leaving friendly bacteria alive. So this is a diagram of a stomach. In your stomach, you have a bunch of friendly bacteria that help you digest food. They're really critical for your function. That's why sometimes when you get antibiotics, you have stomach discomfort and whatnot. It's because you're killing off your friendly bacteria. Now, if you have an infection, you also have some nasty bacteria, some bad bacteria that you want to get rid of. If you're given antibiotics, your antibiotics will just kill everything indiscriminately. Well, not everything, but it will kill your friendly bacteria and your bad bacteria. Phage, on the other hand, you can use phage that are specific against the exact bad bacteria that you're trying to target. So with this treatment, you're actually going to get rid of all of your nasty bacteria while keeping your friendly bacteria alive. So in summary of phage therapy, so clever use of phage therapy can help avoid resistance. And phage therapy can have less damaging effects on the friendly bacteria we rely on. In any case, in the future, phage therapy will be an important tool to combat bacterial infections. It's actually already used a little bit in Europe, but not quite. So the next thing I'll speak of is how can we use viruses to combat cancer? Cancer is a leading cause of death in the United States. So the leading cause of death in the United States is heart disease, and just the second leading cause of death is cancer. Everyone knows of cancer. There's in this sample year, there was nearly 600,000 deaths as a result of cancer. So what is cancer? Cancer is a consequence of uncontrolled cell growth. Let's say you have a normal, layer of skin, for instance, and you have some cells. Every cell knows its place. It knows not to divide too much, and knows just to stay just right where it is. But this skin is exposed to sunlight. Sunlight can actually damage the DNA. It can introduce a change. At a certain point, some cells can, have their restraints lifted. They become cancerous. So, and they can just kind of start dividing as they please. And they divide because they can divide. And over time, these cells can just divide and divide and divide, and they're out of place, and they're invading the tissue. And this is what a cancer is. It's just cells that divide rapidly. So our current cancer-fighting tools are relatively limited. We have chemotherapy, which was pioneered in the 50s and 60s. Now, this is basically a drug. You give a patient a drug. The drug will be distributed around the body, and it will kill rapidly dividing cells. So cells that are dividing fast will die in general. So this will kill the cancer, but it will also hurt friendly cells, like your good cells, and it gives a lot of side effects. Another method we have is radiation. So basically we just shoot a laser of energy at a tumor and just kill all the cells in an area. So if this patient has a tumor here, we just blast it. So what I want to speak about is called oncolytic viruses. Now these are viruses that kill cancer cells, but not friendly cells. So they can be delivered body-wide and to regions difficult to treat with chemotherapy. Sometimes drug delivery for chemotherapy is very challenging. In addition to killing cancer cells, oncolytic viruses can draw in the immune system. So this is just a use of viruses, not to make us sick, but to address an important health concern. So how do oncolytic viruses work? So let's imagine you have some tissue here, and you have a tumor. So these are normal cells around here, and these nasty looking cells are some cancer. You can introduce an oncolytic virus to this tumor. Now, let's say you infect this cell, this cancer cell, and the cancer cell is going to make a bunch of copies of this virus. Now, the cancer cell will die, and the virus will be spread to all cells nearby. The thing about these oncolytic viruses is we have designed them such that they are not able to infect non-cancer cells. So these cells will be protected. These cancer cells will be infected and will die and will spread the virus. And soon you can infect the entire tumor. So in summary, oncolytic viruses can infect and kill cancer cells, but are crippled in normal cells. And oncolytic viruses are an innovative addition to existing cancer therapies. So my take-home conclusions. Viruses don't just cause disease. They can be used as tools. And phage therapy can be critical to combating growing antibiotic resistance. Oncolytic viral therapy is a promising cancer treatment. Any questions?